It appears that the centrosomes and the centrioles coordinate the activity of the spindle fibers. You know, so this is the centromere. This is the centrioles, and this center where all the spindle fibers are kind of coordinated is the centrosome. Make sure that you know this is the centrosome, centrioles, centromere of the chromosome, centromere of the chromosome. So the first division is going to occur. They are aligned at the equator of the cell. These centrioles are at the poles of the cell, and then these fibers, protein made, actin probably, starts separating homologous chromosomes. Separation of homologous chromosomes, and they migrate very close to the poles. This will be a typical telomere. And then the nuclear membrane it gets regenerated. Cytokinesis start making the, how you call it?
Membrane. No separation.
Meiosis.
No, the shape. I forgot what. The cleavage.
The cleavage. You see the cleavage right here, beautiful cleavage, and the nuclear membrane start regenerating there, and there will be a point. This is regenerated. There will be a point where the cleavage finish up the division, and now bingo, you got the two first cells. Now we have a problem here. We cannot make a sperm or an egg with this, because then your babies are not going to be diploid, but they are going to be triploid. And polyploidy is bad, probably will die in years. So we better resolve this, and Mother Nature figured out that the best way of doing it is by making another cell division. And this is what is going to happen. This cell you will see it like in a typical prophase with no organization, right there. And then they will align neatly at the equator of the cell. Do that with that. This is a metaphase of
Phase 2.
meiosis 2. Metaphase 2 of meiosis 2. And they are aligned beautifully in each cell at the equator of the cell. And then the second meiotic division will start, you do that cell. And okay, so you can start thinking about the fibers, the spindle fibers, working out that way. Right here you have the centrioles, and then the spindle fibers will start. This is a typical, right up to right here is a typical anaphase 2. And if they continue migrating toward the poles, then you end up with these chromosomes right here at the poles. The nuclear membrane start regenerating. The cytoplasm of each cell will start making the cleavage, which is the cytokinesis. Right here you have the nuclear membrane regenerating, and the cleavage will end up with the division. And now you have one cell, 2, 3, and 4. These are the 4 cells that potentially meiosis will produce. And how many of them, if in one ejaculation of a boy you have more than 1 million sperms, could you imagine if one meiotic division produces only 4, how many of those will have to carry on in those testes in order to have that volume of sperms? It's amazing. Now, I mention these not because I want you to start thinking about those things, but because the number of divisions in order to equal 1 million are thousands and thousands of divisions, and it's a complicated system. How many mistakes can be done? A lot. Potentially, a lot of mistakes can be done, and some of the mistakes won't be threatening. It's just a variation that is going to accumulate in thousands of years and maybe leading to different traits and abilities and capacity to adapt. There will be gross mistakes where then you will have to deal with a baby that is not very good. But thinking positively, now you have either these 4 eggs or 4 sperms. Is there any one of these that is the same?
No.
Can you see that this process of recombination has worked out? And this is just a pair of chromosomes. Now imagine what would happen with 23 pairs. Gosh, this is getting really complex. And you can start imagining the beauty of organization in those incredible cells that make us up all. But now, imagine that these are products of 2 different individuals, say a boy or a girl. And what would happen if, just by random chance, this is the egg? Well, let's say that this is the egg of the girl, and this is coming through the sperm. And it's coming, find the egg, penetrates the egg, and this will get right there, and this will get right here, and this is a twin cell again. They are not going to join because they are independent chromosomes. They are not copies of themselves. So this is the twin zygote. So this is the zygote, your baby. Now, imagine your wife has all these different genes homologous to these genes that you have in your spermies, and your wife has all these different genes that are not present in your sperm. So now you can see how your babies are going to be resembling you, but at the same time being so different. And no wonder if you have a sibling, then you can say, you know, I have a sibling, but she is so completely different as compared to me. Or a boy, or tall, skin color, everything segregates and varies at this point. You can put another sibling here, and you will see that this sibling is going to have a different composition to this. And right here you have it. Now, this sibling has this component of the father, while this doesn't. And this shorty chromosome has a component of the mother, while this has the component of the father. And is when you say, you know, you resemble your mother because you have more information coming from your mother. Or you resemble your father because you have more red beads. Or you look just like your grandpa. You smile like your grandpa. You walk like your grandpa. And you keep hearing those things as the parents talk about you. You know, and this is exactly that process of, first of all, crossing over. Well, first of all, is chromosome duplication, synapses, crossing over, and then halving of the chromosome number down to from the 2N to 1N, and then carrying out the complete separation of the sister chromatids. And these are your babies now. And but you know, in lecture I'm gonna tell you what is the capacity for this variation to be produced by a human being, and it's going to be in the billions of possibilities. And I'm gonna demonstrate that by simple mathematics and probability. Don't be afraid. Yes. So I don't know if you have any questions. I don't know if you have the courage to do it yourself, or